Everything has two sides, and this is extremely true for photosynthesis. We've all learned that light provides the plants with energy to make their own food, but do you know that light could also cause damage to the plant cells during the process of photosynthesis? So, how does light cause damage in plants during photosynthesis? Because of the large differences in redox potential between reactants and products and the involvement of excited intermediates, photosynthesis poses a unique problem for plants with respect to the generation of free radicals and other oxidizing molecules, which can cause damage to the cells. So, what are free radicals? The key feature of all free radicals is the unshared electrons in valence shell. Here are two examples. The unshared electrons make these free radicals very reactive because they tend to grab electrons from other atoms or molecules to fill their outer energy level. The problem is that when free radicals steal an electron from surrounding compound or molecule, a new free radical is formed in its place. In turn, the newly formed radical then looks to return to its ground state by stealing electrons from other molecules. The chain reaction continues and can be thousands of events long. A broad range of macromolecules and cellular structures can be damaged during this chain reaction, like the lipids shown in the picture. There are many types of radicals, but those of most concern in photosynthesis are derived from oxygen and known as reactive oxygen species. Sequential reduction of molecular oxygen leads to the formation of a group of reactive oxygen species. The structure of these radicals is shown in the figure below and along with the notation used to denote them. Another reactive oxygen species created in the photosynthesis is singlet oxygen. Singlet oxygen is the excited form of oxygen, much more reactive and unstable than the ground state oxygen, and tends to steal electrons from nearby molecules. These free radicals are generated at three major sites during photosynthesis: the light harvesting complex associated with photosystem II, the photosystem II reaction center, and the photosystem I acceptor site. Let's first look at the light harvesting complex of photosystem I. Absorption of light causes chlorophyll to enter the singlet excited state, and the excitation energy is rapidly transferred between neighboring chlorophylls in the light harvesting center. However, before excitation energy is trapped in the reaction center, triplet chlorophyll can be formed from singlet chlorophyll. In contrast to the singlet chlorophyll, triplet chlorophyll is relatively long-lived and can interact with oxygen to produce singlet oxygen. In the photosystem II reaction center, light energy boosts the electrons in the P680 molecules to a higher energy level, and these excited electrons leave P680 to the pheoplatin molecule, the primary electron acceptor. However, the oxidized P680 can be turned into triplet P680, which react with oxygen, generating singlet oxygen. The problem with photosystem I occurs on the acceptor side, where electrons reduce NADP+ to NADPH. However, because the acceptor side of photosystem I has a low redox potential to reduce NADP+ via ferredoxin, the electrons are likely to reduce oxygen to hydroxyl radicals, an extremely toxic type of reactive oxygen species. Because this reaction occurs on the strongman side of photosystem I, the reactive oxygen species can cause damage to key enzymes in the Calvin cycle. Generation of these oxidizing molecules occurs at all light intensities, but when absorbed light energy exceeds the capacity for light energy utilization through photosynthesis, the potential for photo-oxidative damage is exacerbated. Thank you very much for listening to my presentation. And these are the works cited for my presentation.